the previous video, we learned protozoan movement that involves their own cytoplasm to form a pseudopodia. In this video, we will focus on two other forms of movement which involves movement using cell appendages like cilia and flagella. In this video, our focus is going to be on flagella. An example of an organism that uses flagella is Euglena, and you can see this single long whip-like locomotor organelle. So that is what we are going to learn about today. Protozoans that have flagella are called as Mastigophoran. Here mastig means a whip and foreign means bearer. So the primary method of locomotion for these organisms is by using the long whip-like flagella. Other than Euglena, there are other organisms too. So we have Giardia, Leishmania, Trypanosoma, and Trichomonas. All of these use different types of flagella and different number of flagellas for their movement. Eukaryotic cells also possess flagella. One example are the collar cells, which are the characteristic feature of phylum Porifera. These cells, if you notice, you can see the flagella coming out. So it takes the water in, and these flagella reach out into the water to pick out food particles, which then takes it into the cell and then it digests them. Sperm also shows flagellar movement. Not only eukaryotes, bacteria also has a flagella. So Salmonella, Vibrio, and Pseudomonas species, all of these have flagella. You can see that the position of the flagella is different as well as the number is also different. But this flagella is not the same as eukaryotic flagella. Their structure is very different. So we would not be discussing them here. Our focus is eukaryotic flagella. What is the origin of a flagella? Like, where does it come from in a cell? When you focus on where the flagella grows out from the body of a euglena, this is what we see. So the outermost cover is called as a pellicle. Euglena doesn't have a cell wall. Instead, they have this protein coat, which is the pellicle. Inside the pellicle, we find plasma membrane. And just below the flagella or cilia, we find an organelle called as the basal body or the basal granule, kinetosome or blepharoplast. This is the organelle responsible for the formation of flagella and cilia. It is a modified centriole and it is present in the ectoplasm, meaning on the edges of a cell, almost near the cell membrane. And its structure is very similar to that of a centriole. Both of them have a cylindrical body. If you look at the cross section, the cylindrical body is formed by 9 peripheral triplets that are arranged in a circle. The triplets are made of the microtubule protein tubulin. The rings are designated as A, B, and C and named from center to the periphery. Both A and B cross the basal body and they can continue into the flagellum or cilia. So what I mean is A and B becomes part of flagella, but C remains within the basal plate. So what this would look like is, so our basal body has 3 rings, right? So A, B, C. A and B continues into the flagella or the cilia, but C does not. So the basal body triplet at some point becomes the flagella or cilia doublet. This means that the structure of the basal body and the flagella is quite similar. But unlike the flagella, there are no central microtubules. There are connections between the peripheral triplets. The central portion is called as a hub. The peripheral triplets are connected to the plasma membrane by connecting tubules which are called as rootlets. These rootlets also connect the basal body to the nucleus. The rootlets receive signal from the nucleus and it can help to alter the orientation of the flagella accordingly. Now let's look at what the cross section of a flagella or a cilia would look like. It is also made up of peripheral microtubules, but here instead of triplets, we have doublets and there are 9 of them. Not only that, at the center, we have two complete rings of microtubule called as the central microtubule singlets. This sort of arrangement is called as the 9+2 array of microtubule arrangement. These are made of tubulin protein. The A ring is towards the outside. It is small, but it is a complete ring. Whereas the B ring, which is more towards the inside, is bigger, but it is incomplete. The peripheral microtubule doublets are connected by linker proteins called as nexin. A tubule of each of the peripheral doublet bear a paired arm which is called as the dynein arm. They are made up of the protein dynein. Dyneins are protein motor molecules that help in various cellular movements. All the dynein arms are oriented in the same clockwise direction. The central singlets are enclosed by a fibrous sheath called as the inner sheath or the central sheath. The peripheral doublets are enclosed by the outer sheath, which is an extension of the plasma membrane. The basal granule doesn't have the central singlets, therefore it doesn't have the central sheath as well. And this can be seen only within the flagella. The inner sheath is connected to the A tubule of each of the doublet by an elastic fiber. They resemble the spokes of a bicycle that connects the center to the rim. And hence, these are named as radial spokes or radial bridges. These are very important structures because they decide how much these doublets can slide past each other. And that is going to decide how much a flagella can bend during movements. All these structures contained within the plasma membrane together are called as axoneme or the axial filament. The components of an axoneme are embedded in the matrix. Now, there are different types of flagella present and they vary based upon the presence or absence of lateral appendages as well as the number of rows present. The first one is stichonomatic, which looks like this. It has one row of lateral appendage. It is seen in Euglena and Astacia. The second type is called as pantonomatic. Here there are two rows of lateral appendages and it is seen in Peranema and Monas. The third type is called as acronomatic. Acro meaning the end. Here there are no lateral appendages but the terminal axoneme is naked. It is seen in Chlamydomonas and Polytoma. The fourth one is pentachronomatic. They have more than two rows of lateral appendages. They have a naked terminal axoneme and it is seen in Urceolus. Finally, the last one is anematic. There are zero lateral appendages. They do not have a naked terminal axoneme and these can be seen in Chilomenus and Cryptomonas. The flagella also vary in their number. For example, in Euglena, you can see that there are two flagella. One is long and the other is short. In Trypanosoma, there is only one flagella and it is towards the rear end of the organism. In Trichomonas, there are four. Giardia has four pairs of flagella. And finally, Trichonympha have many flagella.